Okay, so in cell cycle, what is cell cycle? Cell cycle means a cell is entering into a cycle of dividing, growing, and again dividing. Dividing, growing, and again dividing. Okay, clear? So, in order for a cell to divide, it must complete several important tasks. Clear? See, understand it like this. If you have a chocolate and you distribute the chocolate in two parts, into children, okay? So you will break it into two equal parts and give it to two different children. So the actual size of the chocolate was bigger, but when you have divided into two, it has reduced in size. Yes, same thing happens when a cell divides. When a bigger cell is there, it divides into two, it becomes a smaller size, smaller cell. So what actually happens? Again, for the next division, if that particular cell, if that particular piece of chocolate, again going back to the example, if that small piece of chocolate which you got from your parent is, again you need to divide it into, you are giving it to your two friends, so you are dividing it into two, breaking it out into two and giving it to your friends. So now what will happen? The piece will become smaller and smaller with each division. Clear? But this should not be case. Again, if you want to distribute the chocolate to your friends, if it happens like this, the chocolate size grows to the original one again and then you divide, there will be benefit to your friends. They will get the same amount of chocolate which how much you received from your parents. Clear? So for every division, the division will be good if the cell, if the chocolate size remains, becomes the same as it was originally there with the parent. Same thing happens with the cells also. When cells divide, they are big cells. They divide into two and they become smaller cells. Then before the next division occur, they should grow in size. They should make their content more. They should have all these cell organelles grown up. Then only they can divide. And when again they divide, they again becomes, give rise to smaller daughter cells, and after that again the daughter cells will have to grow. So this is the cell cycle where a cell divides, grows and redivides. Clear? So for division, this one point is clear. For a cell to divide, it must grow, synthesize the materials which are required, like the proteins, RNA, etc., and duplicates its DNA also, because again DNA has to be distributed. So DNA should be duplicated. It should have duplicate copies of the DNA so that equal amount of DNA goes into the both daughter cells. Clear? Just the splitting is not accepted. It should be equal distribution. Clear? So what actually happens here? Cell perform these tasks in a predictable series of events, and these series of events are called as cell cycle. So to come, if we see the definition of cell cycle, it is, the cell cycle is a series of events that take place in a cell leading to duplication of its DNA and subsequent division of the cell to produce two daughter cells. Okay, the highlighted points, the key points are in highlightation. Duplication of its DNA. What is the need of cell cycle? Two needs: duplication of its DNA and then effect division of the cell. Subsequent division of the cell giving rise to two daughter cell. We generally call it as a daughter cell only. It's not depending on the, it has nothing to do with the male female category. Okay, it's just the depiction of daughter cells. Clear? Don't get confused here. So a cell cycle is a series of events that take place in a cell leading to the duplication of its DNA, and then after the duplication of DNA, what happens? The division of the cell occurs so that two daughter cells with same amount of DNA are formed. The cell cycle consists of two phases: a non-dividing phase, which is called as the interphase. Inter means intermediate. Non-dividing phase is the interphase. You can correlate it with intermediate, like between two division phase, an interphase. Between two division phase, an interphase. Okay. Then the second is a dividing phase called M phase or simply mitosis. Okay. So two phases in a cell cycle, the interphase, which is the non-dividing or the preparation period, and then the dividing phase where actual division takes place. That is the M phase or the mitosis. Mitosis, again recall here, mitosis was being done for what was the necessity of uh, mitosis? Growth, replacement, and repair. Clear? And what is the name of the other type of cell division? Meiosis. In which type of cells meiosis occurs? Sex cells. What kind of division it is? It is reductional division. Okay. Furthermore, coming on to the interphase. So the non-dividing phase is called the interphase. Just see this diagram here, this circle here. The interphase consists of three phases, G1, S and G2. Why at all this interphase is needed? I have already told you. Just again, just a quick revision. The two daughter cells which are produced from the mother cells are relatively smaller. So relatively smaller in size, although they have a full sized nucleus, but relatively less amount of cytoplasm. And when the cytoplasm is less, the cell organelles are also smaller and not in proper content, not in proper amount. So these cells are said to be in interphase. Earlier interphase, when there was when observed when this phase was being observed earlier, there was no change in the chromosomes were seen. So it was being assumed to be a resting phase of the cell. But when you will learn about this interphase, you will come to know that it is one of the active states of the cells. The cell is quite active during interphase. It is not at all resting. Okay. During this phase, the cell is preparing itself for the next cell division and growing in the same size as that of the parent cell, as that of the mother cell. Okay. So in reality, the cell is actually very much active in the interphase. Earlier it was considered to be a resting phase, but it is not. Now the interphase has three phases. The first growth phase, see here the G1 phase or the first growth phase. The RNA and the proteins are synthesized during this phase. What are synthesized? RNA and proteins. The volume of the cytoplasm increases. And the mitochondria in animal cells and chloroplast in plant cells also divide. One important thing here to note is that mitochondria and chloroplast also have their own DNA. It is a very important point, and when further in the next classes you will be opting for biology or studying in greater detail, you will come to know it has a very great significance in identification of a human being, identification, individualization of a human being. So mitochondria in animals and chloroplasts in plants, they have their own DNA in addition to the nuclear DNA. Clear? So during the first growth phase, what is being synthesized? RNA is synthesized, protein is synthesized, and mitochondria and chloroplasts are also dividing. What else is happening here? They may either after these things are being synthesized, they may withdraw from the division. They have to decide whether they have to further enter into the division phase, or now they are just staying whatever size they have attained. Till here they have attained, after the G1 phase they have attained the size of the parent cell. Now after attaining the parent cell size, they have to still decide, the cells have to decide whether they want to enter into the division phase. If quick division is required, they will enter. If new cells are not required at that time in the body, they will not divide for some time. Whenever the need is there for division, then they will divide further. Okay. So let us suppose that they are entering into the next phase. So the next phase here is S phase or the synthetic phase. This phase is called as synthesis phase because here the most important thing, DNA is being synthesized. DNA already the cells have inherited from the cells have received from the parent cells, but here for the division to take place, what is necessary? Duplication of the DNA is necessary. So in the S phase of interphase, DNA synthesis takes place and the chromosomes are duplicated. It's a very important factor. Okay. Now coming on to the third phase. Third phase is the G2 phase or the growth, second growth phase. What happens in the second growth phase? It's a shorter, you can see in the diagram, this chart, that the space occupied by the G2 phase is a very less. So out of these three phases of interphase, G2 is a shorter phase. Here more amount of RNA and proteins are being synthesized. What kind of RNA and proteins are synthesized here? The RNA and proteins which are necessary for the cell division are synthesized in the growth 2 phase. Now after the cell growth, the G2 phase or the second growth phase, the cell is very much ready to undergo the cell division and enter into the dividing phase of the cell cycle. The second phase, that will be the M phase. Clear? After this, the dividing phase is completed by the cell and the new cells are formed. Dividing phase I will discuss later. So till now this much is clear. The interphase has three phases. G1 phase where the RNA and proteins are synthesized and cytoplasm content is being increased. Mitochondria and chloroplasts are dividing. Okay. Then in S phase, DNA duplication is taking place and chromosome duplication is taking place and DNA is being synthesized. And then the G2 phase, growth 2 phase, here more amount of proteins and RNA is being synthesized and these proteins and RNAs are necessary for the cell division. So whatever RNA and proteins which are necessary for the cell division further are being synthesized in the G2 phase. Clear? Now, the next. Before we start up with the division phase, there is one more important topic here, that is the formation of new DNA. During the S phase, what is happening? The DNA is being synthesized. How the DNA is being synthesized, that is important here to note. Just see this diagram. Here you can see a DNA double helix. This structure I have told you. This is a DNA double helix. During the DNA replication, synthesis, we call it by a scientific term DNA replication. So when DNA is replicating, it is unwinding. You can see here the two strands are unwinding, they are separating. The parent strand is separating into two strands. Blue color one is the parent strand. Now one parent strand is opened this way and the other is going that way. Now every parent strand is getting a complementary strand synthesized, which I have denoted here with a pink color. Can you see? The blue one is the parent strand, whereas the pink one is the new strand which is being synthesized on the parent strand. And again, I told you in the previous class, it is because of the complementary base pairing. Adenine will be forming two hydrogen bonds and joining with thymine, and guanine will be forming three hydrogen bonds and joining with cytosine. Same complementary base pairing occurs here also, and then they are forming the new strand on the template. Template, you understand? Template of the old strand. Clear? Coming on to another diagram. See here, I have depicted it more clearly here. This is a parent strand. The first diagram, this here, the parent strand double helix. Here, the parent strand is opening. It is opening in this direction, and when it is opening, the new strand is continuously formed on the old strand. See here, the movement. Further, it unzips, it opens, it uncoils, and the synthesis, the replication of the new strand progresses in this direction, and then we have got two newly synthesized DNA strands which have complementary. That means here one strand of parent and one strand which is newly synthesized